In a previous lesson, we learned about the reactivation hypothesis, that bits of the brain that are involved in processing the perception of objects are also reactivated when we imagine those objects, suggesting a role for memory in that same place. And we saw evidence from fMRI data that perceiving a face and imagining a face both activate the fusiform area, although the imagery shows a weaker signal. We had lesion data supports the reactivation hypothesis. People who have damage to this part of the brain, they report color blindness, but they also can't imagine colors. It's as if they can't reconstruct images with color, because that same part of the brain is necessary for color memory, as it were. And early stimulation data stimulating visual areas produced visual memories, stimulating the auditory area produced auditory type memories, all supporting the reactivation hypothesis. Now, in this study, scientists are going to be recording from cells in the hippocampus. And we previously learned that the hippocampus is a critical memory structure. It lies underneath the temporal cortex. It's the gateway to the long-term memory system. So earlier we learned about the working memory system. This is a short-term, limited capacity memory system. Whatever we're sort of thinking about or perceiving or whatever's going on in our mind at the time, that's the content of our working memory. But whatever's in working memory has the potential to be written to the long-term memory system, and the hippocampus and surrounding tissue seems to be critically involved in that process. Now, in this study, then, scientists are going to be recording from cells in the hippocampus. Earlier, we used this picture to show what it's like to do an experiment where you're recording from cells in the fusiform face area. So this cell here would be in the fusiform face area, and what scientists do is, in an awake, conscious person, the person's looking at a computer screen, and then different kinds of stimuli are flashed on the computer screen, and scientists simply record electrical activity of a single brain cell in this region. Well, as it turns out, cells in the fusiform face area have faces as their preferred stimulus. So when a face is on the screen, you get maximum activity, electrical activity of the brain cell, compared to, say, an object or a scene. So in this study, though, scientists are going to be recording from cells in the hippocampus and the nearby surrounding tissue called the entorhinal cortex. So here's the experimental paradigm. We have the subjects, and again, there are electrodes in their brain. They're effectively watching, a computer screen, and they're going to be watching 5 to 10 second clips. And each clip, you can think of it sort of as an episode. Might be famous people or landmarks or the introduction to a TV show or something. So just short little video clips. And they'll see a whole bunch of these little video clips. And then there's an intervening task. So the subject, for 1 to 5 minutes, does some other kind of activity. And then after that, they're going to be asked to recall what they remember seeing during the viewing session. Okay, what do you remember seeing? No other external cues, and most of the subjects could remember what they saw just minutes earlier. Now remember, they're recording from cells in and around the hippocampus. So what in the initial viewing session, as the subjects are watching scenes, they want to try to find cells, or they're just hoping that the cell they're recording from will respond to one of the video segments. And sure enough, they found cells that seem to selectively respond to one out of the set of video segments that they played to subjects. So the data you're about to see here would be the data from a single brain cell that has a preferred stimulus, one of the video clips. And the interesting thing was whether that same cell would become active when the subject recalled the video clip. If so, that would support the reactivation hypothesis, that this cell was active during the initial processing of the video clip, but also when the subject was retrieving the memory of that video clip. So let's take a look at the data. Okay, so this will give us a view of what the experiment involved. The person would be seeing what you see in this region of the screen here, and then down here will be a record of the neural activity of one cell that they're recording from. And if there's just a small signal down here, the cell was not firing very much. It was not electrically excited. And here we're going to see that the cell gets really excited, and we'll try to match that up with what the person was seeing at that time. So here we go. So time is moving on. that one brain cell doesn't seem to be responding very much. It's not responding very much to Dr. King or to whatever this is. Here's a scene from Titanic. That brain cell doesn't seem to care much. Oh, The Simpsons. So we get a burst of neural activity when the Simpsons segment came on. Back to the Dr. King, the neuron quiets down. Brain cell not very interested. Scene of the pyramids. Sex in the City doesn't really activate that brain cell. Marilyn Monroe. Not much. Here we have the scene from Titanic again. Again, the cell doesn't seem to respond very much. Dr. King, no. And you get, what do you think's going to happen here? It's going to be The Simpsons. So what we've got here, by the way, that's Itzhak Fried, and it's his team, his research that we're looking at here. So what we have now is that Itzhak Fried and his team have identified a brain cell that seems to selectively respond to the Simpsons clip and not the other video clips. So when the subject sees The Simpsons, that particular brain cell became highly active. The question is whether that same brain cell will be reactivated when the subject remembers or recalls the previous clip that they just saw. So now we're in the recall session. So you'll see what the person's going to say here, and then again we have the recording from that same neuron. Now we know this particular brain cell responded to The Simpsons, so we're going to see whether that same brain cell will be reactivated when the subject remembers seeing the Simpson clip. Okay, so let's see what the first memory is going to be. Something about New York, maybe the Wall Street scene. But notice the brain cell did not respond. The Hollywood sign she remembered seeing, but the brain cell didn't respond very much. Boost of activity, The Simpsons. And so here we have direct evidence for the reactivation hypothesis, that the very same cell in the entorhinal cortex, which is a region near the hippocampus, that very same cell that was active during the perception of the Simpsons clip was reactivated when the patient was recalling, remembering having just seen the Simpsons clip. So when the person is recalling the Simpsons clip, that cell is involved in triggering the memory, reactivating the cells that were involved in processing the clip to begin with. And so then whatever cells become reactivated, they then generate the content for working memory, and the person has a recollection. They remember seeing The Simpsons, and that's what their working memory content would be. Now, how vivid that is may vary between individuals, but nevertheless, the basic concept is that the cells that were used to process a stimulus are reactivated when a person remembers that stimulus.